Thank you, Johan, for this very kind introduction, and I'm very honored to be part of this distinguished set of faculty members and speakers. So in the next minute or so, I will address, unlike the polygenic or multigenic subtypes of psoriasis that we heard about this morning, mostly about what is assumingly dependent on the single gene variant or models of psoriasis or psoriasis-like disease. And it has been long thought out that the pustular variants of the disease were suitable for this model based on some very basic clinical features that I will review. These are my disclosures. So the genetic architecture before the first evidence of the first identification of the first pathogenic genetic model of pustular disease, which is not IL-36 RN, but which is historically CARD14. It was thought that some pustular variants of the disease might be related to a monogenic Mendelian model. Early onset of the disease, the sort of big blowout of sort of peaking inflammation, well, with a massive inflammatory cascade, and also not only the early onset in childhood or sometimes in the neonatal period, but also in some cases some pedigrees with or without relatedness suggestive for an autosomal recessive or autosomal dominant Mendelian model. So we will speak mainly about this model and discussing the intersection between, if any, between pustular disease and plaque psoriasis. The reason is that, whether you like it or not, according to the latest national database surveys in Sweden and in Taiwan, also in France, for instance, for generalized pustular psoriasis, 40 to 50% of these patients, they have associated plaque psoriasis. And this remains as an epidemiological mystery. And I think this cloud of mystery will remain after this talk, but we might discuss about it. So I mentioned it, the first group being able to identify the first causal pathogenic mutation of any gene in pustular disease is Anne Bocock's group in 2011. That was part of the study in both familial and sporadic cases of plaque psoriasis with or without psoriatic arthritis. But there was also one individual of undebatable generalized pustular psoriasis in an infant. And since then, a lot of heterozygous or more seldom homozygous or compound heterozygous mutation, gain-of-function mutation of a gene named CARD14, so caspase-associated recruitment domain 14, and have been evidenced by several labs. You can see on the upper part of this slide, for instance, oops, sorry about that. Here, for instance, in red, some genetic mutations which have been associated with both psoriasis vulgaris, pityriasis rubra pilaris by the group of Alice Prescher and Yunewito, and generalized pustular psoriasis, showing that, and this is the same variant, meaning that there should be an intersection between the different disease, although we don't know what is the transcriptome of the lesional skin of these different disease belonging to the psoriasis spectrum. And you can see that there are some variants that mainly lie in the sort of hot spot, mainly in the CARD or coil domains. And this is accounting for 60 to 70% of the mutation identified so far. Some are privately associated to psoriasis vulgaris, again with or without psoriatic arthritis, and some are associated with PSA. But you can see that again, there are some mutations associated with psoriasis vulgaris and GPP, and others with GPP only. On the right-hand side, you can see patients that we are following and who's fitting with the description of psoriasis vulgaris, and you can see the orange soles, and she had also salmon-like coloration of the face, fitting quite well with the description of pityriasis rubra pilaris. So that means that for the aficionados of the hermetic cleavage between PRP, psoriasis vulgaris, but also GPP, I'm sorry to disappoint you, but there should be some intersection between this disease, and probably the disease, the clinical presentation are influenced by other factors. It could be epigenesis, it could be environmental triggers, or disease-modifying genes, or among others. So one important milestone in this context was the capacity of some group to reproduce in mouse some conserved residue in human and mouse, to reproduce the mutation identified in humans in mouse. So the mice carrying the heterozygous mutation, the CARD14, especially the 138 residue, especially the deletion or the substitution of the glutamic acid in position 138. Two groups in China and in Switzerland, last French in Switzerland, were able to identify the capacity of this knock-in model in mouse to reproduce psoriasis-like disease with the presence, as you can see here, I try to touch, but with delicacy, here, the presence of intraepidermal microabscesses of neutrophils, which are immunostained by the LY6G antibody specific for neutrophils on the right, in the middle part of this cartoon. And what was shown in these papers is that there was the impact of this knock-in, this mutation, heterozygous mutation, was enhancing the response of keratinocytes to IL-17, polarizing the inflammatory cascade towards an IL-23, IL-17 pull, if you wish, and that the mobilization of the protein of KARMA2 was associated with a physical association with ACT1 and TRAF6 as being transducer of the IL-17 receptor signal. So these gave some kind of credibility to the causality, of course, of this heterozygous mutation, and also gave some quite clarity about the rationale, how the mechanistic scenario is installed. First, Anne Bocock set the scene, showing that the CARD14 gain-of-function mutants, some of them, they were enhancing the engagement on NF-kappa B following the BCL10 and MALT1 engagement, and also upregulated, as you can see on these slides, the transcription in keratinocytes of IL-36 gamma. So there is a connection with IL-36 between CARD14 and IL-36. But what was also very clear is that the Chinese paper and the paper by Lys French also provided some rationale for the privileged response to the IL-23 and IL-17 blockers of what has been called as CARD14-associated papillosquamous eruption, a CAPE, but also of the cases of psoriasis vulgaris associated with CARD14. So you'll tell me, I mean, this is canonically engaged, IL-23 and IL-17, at the population level in psoriatic disease. But these patients, they fail to respond optimally to TNF blockers or other targeted biologics. So it really gives some credit to again, this IL-17 dependent scenario caused by CARD14. The imiquimod mouse model, which you know about, is CARD14 dependent, but we will see that it's also IL-36 receptor dependent. So there is still a cloud of mystery in the imiquimod mouse model. The second set of genes that are those related to the IL-36 pathway, so I will go quickly through this. Two groups in 2011 reported, including ours, reported the homozygous or composite heterozygous mutation of IL-36RN, a gene encoding for the IL-36 receptor antagonist. All these patients fulfill the criteria for generalized pustular psoriasis as a model of acute intermittent inflammation, some of them having some chronic inflammation at a very specific location. You can see all these patients, they have the same so-called L27P mutation, and you can see there is acropustular lesions being chronic. There are also geographic or fissuratic tongue, flexural lesions, or plaque plus pustules. So the disease is not that homogeneous as we would expect. And at that time, the group of Jenny Towney and John Sims in Amgen R&D provided the mouse model of this, combining the homozygous invalidation of the IL-36RN gene in mouse with the skin-directed transgenesis of the IL-36 alpha encoding gene, leading to a psoriasis-like disease with the presence of neutrophils, but not of macroscopic lesions with sterile pustules. So since then, a lot of groups have evidenced IL-36 mutations with the frequency varying from 5 to 70%, depending on geographic area, Asia being probably the continent where the IL-36RN mutations are the most prevalent. And also for, again, those of us who are still strong believers of the hermetic cleavage between GPP and AGAP, the same mutations have been evidenced in patients with GPP and with AGAP. So, and we'll see that with infectious triggers being a trigger for GPP, it makes quite complex to differentiate very clearly AGAP to GPP in some cases. So this is from a paper we published 7 years ago, showing that there are some new mutations without any, this is basically Western blot assessing the expression of IL-36 receptor antagonists. So we reproduce by site-directed mutagenesis the mutations of IL-36RN genes that were in the public domain. And after transfection in human embryonic kidney cells, we're able to show that on the left-hand side, the red box, you can see that there is no protein expressed, showing that these are really null mutations, usually associated with severe phenotypes, so not really localized, but GPP or AGAP. And on the right-hand side, you have so-called hypomorphic mutation with a lower expression, but still existing. And on the bottom of this side, you can see a functional test, which is a luciferase assay with the NF-kappa B promoter reporter gene in cells expressing both the IL-36 receptor and transduced with the IL-36 gamma encoding constructs. And you can see that some of these, in this luciferase assay, the wild type basically is working very well in reducing the luciferase activity, but some of the variants, they don't seem to do anything on the repression of the inflammatory cascade, where others are clearly associated with the total absence of any effect. Another very interesting finding is also that there are some patients where the expression of IL-36 receptor antagonist is intact, but the functionality is absent. And that's the case for these very specific mutations, unraveling the key role of the cleavage of the immature form into the mature form of the antagonist. In this case, there was a missense mutation in position 2, where the substitution of a valine by a phenylalanine resulted in the retention of, shown by mass spectrometry. That's basically, yeah, the mouse pad is very sensitive. Yeah, it's not working. You can see the blue line here. Which shows the power of the wild type in reducing, again, in the luciferase assay, the activity of the IL-36-dependent IL-8 luciferase construct. So basically, the antagonist is working quite well. And the V2F construct, you can see, which is in red, doesn't work at all. And on the right-hand side, you can see that mass spectrometry on the lower part of the slide, there is basically a retention of the high molecular weight form, showing no cleavage in the presence of the cleaving enzyme of the mature form of the IL-36 receptor antagonist. And this is due to the fact that this substitution in valine 2 is leading to an impairment of the activity of elastase, which is mandatory for the bioactivity of the IL-36 receptor antagonist. And this is why this elastase, which is a protease very importantly released by neutrophils, it's important to have this cleavage step to activate the antagonist. Last but not the very least, the integration of some external triggers allow us to show that what is dominant in deficiency in IL-36 receptor antagonist, one of the dominant triggering events are the infectious triggers. And it's not streptococcal infections, but it's viral pharyngitis as a very powerful double-strand RNAs from viral origin, a very potent trigger of toll-like receptor and the IL-36 pathway. And you can see on this slide a SARS-CoV-2 induced a flare-up in a patient with a V2F mutation. And what you can see on this nanostring evaluation of the transcriptomic landscape in the whole blood is that there is not so much interferon signature gene that you would expect based on some previous work by Francesca Capone, but as expected, this is dominated by a neutrophilic signature. The neutrophilic signature is more prominent. So again, other genes associated with pustular psoriasis, you can see the list on this slide, and this is an evolving picture, CARD14, AP1S3, SERPINA3, MPO, all these are more or less connected to the IL-36. It doesn't mean that the IL-36 is really the main checkpoints. There is some redundancy and some parallel inflammatory cascades. And again, the prevalence based on this nice paper by Sophie Twelves and Francesca Capone varies, but in this nice cohort, you can see that the IL-36 mutations that target roughly 20% of the patients with GPP. So GPP, yes, is genetically distinct from plaque psoriasis, you can see on this slide. But there is some connection. IL-36, we've seen this morning with Johan Gudjonsson, that the inflammatory players are upregulated in the lesional skin of plaque psoriasis. IL-36RN is not mutated in patients with plaque psoriasis, but I showed with CARD14 that there is a genetic basis for the overlap between the two entities in some cases, although the prevalence of the CARD14 mutant is not that big. So this cloud of mystery remains. What is the big connector for pustular psoriasis and plaque psoriasis? This applies also for palmoplantar pustulosis. We don't know. Probably other genetic factors. There might be other players, but surely some new genetic determinants favoring this time instead of IL-36RN, more like a CARD14, but the association of these different entities. And also this should invite us to reconsider the disease taxonomy, clearly shifting the pustular psoriasis associated with monogenic models to the family of autoinflammatory disease, meaning a group of disease depending mainly on the innate immune system deregulation and where the adaptive immune system is very accessory. And the other, of course, asset of having this genetic identification of the causal mutants is that if you get lucky from time to time, you have some pharmaceutical industry partners that are able to reach these wonderful milestones with the approval of an anti-IL-36 receptor antibodies, and other drugs are in development with imsidolimab. So spesolimab is development is a great success. Congratulations to our friends from the industry. So in summary, the identification of a genetic architecture underlying pustular psoriasis and psoriasis-like disease reveals a key contribution of inborn errors of the skin immune system and the systemic innate immune system. The single gene model, yes, may also apply to non-pustular clinical variants, including psoriasis vulgaris, for CARD14, not for IL-36RN. And these single gene models allow the design and further approval of targeted therapies. And with this, I would like to acknowledge all our collaborators, mainly Asma Smahi, Elodie Bal, Marie Tauber, and for now, Florence Hassan, with all the national and international collaboration. Thank you.
Thank you, Hervé, for a great talk. We have time for a couple of questions. Klaus?
Where does the TNF-alpha-induced pustular reaction that you see in the pulmonary cells, where do they fit in? And the paradoxical psoriasis that we also see in rheumatoid arthritis, patients treated with the TNF-alpha blocker?
Great question. We have studies ongoing. As you know, I mean, there is a type 1 interferon, strong type 1 interferon signature, including in some pustular cases, in paradoxical TNF-alpha inhibitors-induced reactions. I suspect there would be some genetic predisposing factors yet to be identified, orientating or favoring the emerging of type 1 dependent inflammation in some patients where the regulatory role of TNF-alpha at low dose, kind of calming down the type 1 interferon, especially in PDC, but not only in PDC, would be basically leading to a type 1 interferon-dependent inflammatory cascade. But that's my guess. But these patients are not mutated. If that's your question, they are not mutated for CARD14. They are not mutated for IL-36RN. Crystal clear.
I got one question for you, Hervé. So how, with this mutation that we know, the 5 of them, how big of a proportion of the cases of GPP do they actually explain?
It looks like an easy question, but it's not. Thank you, Johan. No, the reason is that there is still debate around the causality of the heterozygous mutations. I mean, in the Mendelian pedigrees, depending on IL-36RN, the heterozygously mutated individuals, they don't have any phenotype. So we found from time to time, and there is in the Francesca, or the Sophie 12 paper, there is evidence of a dose effect of the presence of there is increased prevalence of the heterozygous IL-36RN mutation in this context. I would say that a reasonable figure in Western countries would be around 20% max, and we have genotype significant cohort of our colleagues from southern Tunisia, around 100 individuals, and only 50% of them are mutated for IL-36RN, none for CARD14, none for AP1S3. We are tackling MPO and SERPINA3, but they don't seem to be mutated. There's still a lot to decipher. Yeah, I think that's a great question.
Great. Thank you, Hervé.
Hervé, just one little question.
Sorry. Yeah, and a short answer.
A very short answer.
Yeah,
what about pustules that occur on plaque psoriasis? And this is what we call psoriasis with pustules. We see this a lot in our population, especially. They don't develop into GPP or even, but they get those weird pustules on the edge of the lesion, and they sometimes, what are those? Where are those in the, sorry.
Well, the current well, official statement is that this is not R36RN-associated pustular psoriasis, but maybe you notice that there is one image from the mutated individuals from southern Tunisia. It's a plaque with pustules on this plaque. So again, I think it's about being, and I'll be quick, Johan. And it's about being rigorous in the analysis of the clinical phenotype, but getting ready and being open based on the existing literature that some heterogeneous phenotype might be present and visible. That's clear. Thank you.
That's great. Thank you.